People from Aachen.
Die sind auch im Gesicht. Frau Heinzelach steht draußen.
Okay. Aber die kommen aus Aachen.
Thank you, Tilman. I'm going to have to try this when I go home and try this knocking, but I may get some funny looks, unfortunately. But it's fun when you travel somewhere new and you learn the new customs. It's one of the special parts of traveling. So today, what I wanted to talk about was the links that Tilman outlined between vascular cell signaling and fibrosis. And in fact, traditionally, I've been trained in portal hypertension and to study the effects of fibrosis on the blood vessels in portal hypertension. But now, in fact, some things are turning upside down and trying to understand how the blood vessels may be playing some role in the development of fibrosis and cirrhosis as well. And I know it's a mixed audience. Some of you are quite expert in the field, and some of you probably want a more general overview. And so I try to keep some things that will be both ways with some cartoons, and also keeping some deeper signaling also. And also I know that some of you are working in cancer, and so the back end of the talk will also have some link-ups to cancer as well. So the question about the blood vessels and cirrhosis really comes to whether blood vessels are required for fibrosis. And there's some evidence that with cirrhosis we saw some of the prominent angioarchitectural changes. You see lots of changes in the microcirculation using some of these cast structures and electron microscopy. So does the blood flow into the liver play some role in this development of the scarring and the fibrosis in liver? And in fact, some people have already studied this. This is some micrographs from the paper from Manuel Morales Ruiz on using, in this case, I think it was sorafenib, or it was one of those related compounds, the receptor tyrosine kinase inhibitors, and showing this anti-fibrotic effect in association with an anti-angiogenic effect as well. And this one was with sunitinib. And again, raises this concept that if you block the vascular structures that change in the process of fibrosis and cirrhosis, can you also reverse the fibrotic process? But I have to mention that although some studies have shown this does occur, David Brenner also had the study with an anti-Tie2 and blocking the angiopoietin pathway that had an anti-fibrotic effect. Detlef Chupin, who is one of your countrymen who is in the US, he showed that actually if you use the wrong angiogenic targets, in fact, things could get worse. And I would ask you to see his paper as well in hepatology, which targeted an integrin and actually showed an exacerbation of fibrosis. So it's a complex interaction. And what is the basis of the interaction? Well, let's think about it. I don't have the answer, what's the basis of the interaction, but we have some hypotheses and some ideas. So of course, the stellate cell is a key role here. And although many of us think of the stellate cell predominantly in its matrix deposition and fibrosis effect, many of us also think of the stellate cell as a vascular cell, and in a way, a vascular pericyte. And so what do pericytes do in the vessels? Some of you who are in the cardiology department know that pericytes are very important in vascular structure. For example, pericytes can release angiogenic factors, and this can recruit endothelial cells and thereby drive angiogenesis. Additionally, some people think that pericytes are also important in the vascular sprout and vascular maturation. So it plays a very important role in angiogenesis. So that leads us to the next question, is what is the relationship of angiogenesis to fibrosis, and what would be the basis by which angiogenesis could lead to fibrosis? Well, again, this is really right now at the level of speculation, but there's some different theories that remain to be tested. One is that is a myofibroblast, and it's tumor and it's desmoplasia like a tumor. And we think about in tumor how very metabolically active cells that are doing a lot of things need blood flow, and without the blood flow, they start to starve. So is there some analogy between myofibroblasts and the tumors? Another is that whether endothelial cells may be some kind of an engine that release cytokines and chemokines, and could this be contributing to stellate cell activation and the ability of stellate cells to be recruited and form fibrosis? So these are a couple of the different possibilities. And where do these cells come from, the hepatic stellate cells? Well, this is an area of a lot of controversy. If some of you who are at the last liver meeting know that although I described the hepatic stellate cell really as a pericyte, there's some controversy here. This is some people think that these are mesenchymal origin, which is probably correct. There's some data from the groups in UK showing that bone marrow vascular progenitors may lead to stellate cells. And then there was some information that they may have some neuronal lineage. Although most likely they don't have a neuronal lineage, they clearly have expression of a number of neuronal markers. And this is, I mention this for a reason, because the protein that I will talk about today in more mechanistic depth is really a neuronal protein. And I'll show you that. That's called neuropilin, and that's what really will be the focus of some of the science that I show you today. Now, regardless of where this cell comes from, the most relevant factor once the cell has an injury is really its transformation to a myofibroblast. And this is the phenotype that leads to more matrix deposition, more angiogenic effects, and more recruitment. And in fact, whether some of the different myofibroblast subtypes have more of these different effects, some more angiogenic, some more fibrotic, that's again a hypothesis that remains to be tested. So really the focus on neuropilin came from a number of studies that were previously published, but didn't really focus on neuropilin. And this was really when we looked in some of the microarray profiles that were done by David Brenner many years back, where he looked at a number of different genes that were upregulated in cirrhotic hepatic stellate cells with in vitro or in vivo activation. One of them was neuropilin. And of course, that was in a list of many, many. And as we discussed earlier, when you have microarray data, we need to confirm it and corroborate. And indeed, this was a protein that's very important in endothelial cell function. And we knew that from our vascular upbringing. And so we looked in hepatic stellate cells. And what we found was that neuropilin was markedly upregulated in both in the in vivo models of liver fibrosis, such as the carbon tetrachloride model here, as well as the bile duct ligation model, and seem to correspond with upregulation of PDGF receptor beta, which also corresponds with the activation of the hepatic stellate cell. And in fact, if you look and do some co-localization analysis of the hepatic stellate cells and myofibroblasts in the cirrhotic liver, I'll show you here on the bottom, you see that there's very close approximation of cells within the scar that are expressing neuropilin as well as PDGF receptor beta with a very high correlation, suggesting that these activated stellate cells are expressing neuropilin. And indeed, in vitro as well, if you look at neuropilin regulation with culture-dependent activation in vitro and just leave the cells in the dish, you see that there's basically a time-dependent upregulation of neuropilin in the, this is in murine hepatic stellate cells, consistent with what I showed you in vivo in the in vivo activation of stellate cells. So what is this protein neuropilin? It's expressed in a number of different cell types, actually. It was first described in neurons, and it's thought in some ways to be a receptor for other chemotactic molecules, such as the semaphorins, which play a role in neuronal axonal guidance. In the endothelia, neuropilin is thought to be a co-receptor for VEGF. And so it, in some studies, suggests actually that it may be the dominant receptor for VEGF responses in vascular endothelium. And for sure, it's a co-receptor whereby it promotes VEGF responses. In cancer, people have also found its expression and suggest that it could be promoting tumor growth. And then lastly, as I show you in stellate cells, we're going to show you some data relating to its ability to act as a co-receptor for a number of the receptor tyrosine kinases as well as serine kinases that are important in the process of hepatic stellate cell activation and angiogenesis. So now I'm going to show you one of the first data sets in some detail relating to the role of neuropilin and how it regulates PDGF-dependent stellate cell motility and chemotaxis. So if you take stellate cells and if you perform traditional gain and loss of function approaches with neuropilin, what you can see is that when you knock out neuropilin function, as shown in panel A, that there's an attenuation in PDGF-dependent migration. And this can also be shown if you track individual cells. So some of you who have some of the cell biology programs that can do this can allow you to track the movement of each individual cell. And you can see that again with the neuropilin siRNA, there's an attenuation in actual cell motility over time. And conversely, if you overexpress neuropilin, then you see an increase in cell migration in response to PDGF. And this, again, is you can use different assays, including the wounding assay. And similarly, with a traditional Boyden chamber assay, where you look at cell migration, you see that if you overexpress neuropilin, here in this case using a retrovirus or an adenovirus, that you can increase chemotaxis, which is what you're measuring with the Boyden assay. So clearly, neuropilin promotes PDGF-dependent motility and chemotaxis. So we can also take an alternative approach. This is a genetic approach where we use mice that have a floxed neuropilin-1 allele. And if you isolate stellate cells or mouse embryonic fibroblasts from these mice, and then you transduce them with adenoviral Cre, which allows you to knock out the neuropilin from the cells, again, you see a reduction in migration, just using an alternative approach with a genetic murine model. So how does neuropilin regulate this process, and what's some of the signaling pathways involved? Well, this led us to a hypothesis here to test, is that I showed you in some of the earlier slides that when you overexpress neuropilin, not only you see the key dominant band, but you see a larger molecular weight form. And that's actually a glycosylation product. And I'll show you some data later on where we mutate that, the site, the serine where the glycoprotein binds, or where the glycosylation attaches to the protein. But this led us to this hypothesis that maybe the glycodomains of neuropilin may act as a sort of molecular net. And this net may allow us to catch a lot of the ligand that's floating around by the cell and allow it to get to closer approximation to the PDGF receptor. And then this approximation and increased binding may promote cell motility. So indeed, if you do scattered analysis and kind of a more detailed biochemistry using iodine-labeled ligand, you can get your binding curves to look at how much PDGF ligand is actually binding to the stellate cells. And what you see is that if you overexpress neuropilin, you see a reduction in the KD, showing a greater affinity for binding, and some modest increase as well as in the Bmax, the total amount of binding. So this suggests that neuropilin enhances the binding affinity of PDGF ligand for its receptor. And so the obvious question there would be, well, is the PDGF ligand actually binding more to the PDGF receptor, or is neuropilin actually acting as a primary receptor that can bind PDGF ligand? Most likely it's the primary. That certainly seems to be the dominant process, whereby if we immunoprecipitate PDGF receptor and see how much of this iodine-labeled PDGF ligand binds, you see that the dominant amount of binding is with the PDGF receptor. If you overexpress neuropilin, you do see a small amount of PDGF, iodine-labeled PDGF binding, but I think that that's probably overall a relatively smaller component, so it allows us to favor sort of the co-receptor model rather than neuropilin acting as a primary receptor for PDGF ligand. So the next question we asked was what's happening intracellularly? So what I showed you previously suggests that there's a model whereby the neuropilin is promoting ligand binding. But in fact, intracellularly, there's a number of features going on that are consistent and also some unique aspects as well. So what you can see here is that, again, if you overexpress neuropilin in hepatic stellate cells and then look at the canonical responses to PDGF ligand, you see things that you expect. So you see increased PDGF receptor phosphorylation, you see activation of AKT, MAP kinase, and with neuropilin overexpression, one of the prominent things you see is increased receptor phosphorylation. And that's expected because you've increased the amount of ligand that can bind with the receptor. But what about downstream? And you'll notice that a number of these pathways don't change. So when you overexpress neuropilin, in fact, there's really no further potentiation of AKT or MAP kinase. But one thing that we did see was a further increase in RAC activity. And RAC, as many of you know, in Journal studies, many of these small GTPases, is very important in cell motility. And in fact, when you overexpress neuropilin, you see an increase in PDGF-induced RAC activation over a time course. And in fact, in some of the experiments, we also saw a very prominent basal activation of RAC in cells overexpressing neuropilin. And if we expand this functionally beyond the biochemistry, you can see that overexpression of neuropilin, again, increases cell motility, as I showed you previously. But importantly, if you use a dominant negative RAC, you can attenuate this process. And so this suggests that activation of RAC by PDGF is potentiated by neuropilin, and that when you block RAC function, you turn off this response. So how does this happen? How do you turn signals that have been activated and potentiated by neuropilin? How does this get selectively to RAC, but yet we didn't see further activation of AKT? Well, that was a question that we wondered. We didn't have any very easy answers. And then we identified this protein c-Abl, which we were actually quite interested in from a portal hypertension perspective in endothelial cells. And we read a lot of the literature showing that this is a non-receptor tyrosine kinase that can mediate a number of signaling pathways from the plasma membrane into downstream signals. And so we investigated this further in terms of the potential role of c-Abl as a transducer, especially since neuropilin itself doesn't actually have any intrinsic kinase activity. But it does have this C domain, which I will talk more about, and I'll show you some data with mutations of this C domain, which is thought to act like an intracellular signal transducer, although it doesn't actually have any kinase function. It can bind other proteins like GIPC and interact with PDZ domains. So here I'm going to show you a number of different blots, and then I'll just summarize the information for a bigger picture. We did a number of experiments, again, using siRNA as well as genetic-based knockdown. Here we use mouse embryonic fibroblasts because these mice that lack c-Abl and its homologs arginine, C-arginine, they are basically perinatal lethal. So we take mouse embryonic fibroblasts from these because we're not able to get stellate cells from them. And what you see is that essentially these cells have essentially no RAC activity in response to PDGF, and they do lack c-Abl as would be expected. And furthermore, if you take LX2 cells and use a complementary approach, which is c-Abl siRNA, you essentially see the same thing that I showed you in panel A, except this is in using siRNA-based approach. And again, as expected, there's very little c-Abl activity once you overexpress the c-Abl siRNA or transfect with c-Abl siRNA. And then if you overexpress neuropilin, even then, you don't see very much RAC activity in these mouse embryonic fibroblasts, suggesting that c-Abl is indeed downstream from neuropilin and can turn off the potentiated RAC activity in response to neuropilin overexpression, but in these MEFs, you don't see that. And furthermore, if you do a few controls, like if you take MEFs from neuropilin knockout mice or c-Abl knockout mice, again, you don't see the c-Abl activation that you see in response to PDGF in LX2 cells. And lastly, how is this occurring? What's the basis? We're still investigating this further right now, but what the information we have to date suggests that there is a co-precipitation between neuropilin and c-Abl, whereby if you immunoprecipitate c-Abl, you can pull down neuropilin, and that this is particularly in response to PDGF. I don't have the group without PDGF on this slide, but this was potentiated when you treated cells with PDGF. So this led us to a model here, and I'll show you kind of this schematic model where PDGF is enhanced in its binding to its receptor by this glycosylation net here from neuropilin. And once it binds its receptor, it leads to signals that lead through RAC1 and c-Abl. And this leads to increased motility, increased migration of the hepatic stellate cells. And this, in turn, leads to more collagen deposition. Now, the question is, can we neutralize this pathway, or is there some clinically relevant mechanism to block this? And so one can envision that neuropilin neutralizing antibody could potentially block this pathway and thereby prevent this process. And so once this last antibody binds, I'll show you the data. So this is the information with the neuropilin neutralizing antibody. And what you can see here, again, a little bit complicated slides, but I'll just point you to the main factor, is that when you use neuropilin neutralizing antibody, you can block in vitro the cell migration that's observed say, for example, in carbon tetrachloride, hepatic stellate cells isolated from carbon tetrachloride animals where they have enhanced motility, enhanced migration, this is blocked by the neuropilin neutralizing antibody. And this is similar if you look at tube formation. Janelle showed a lot of data earlier with tube formation of stellate cells and endothelial cells. And you see essentially conceptually very similar information, since migration is one of the key factors in eventual angiogenesis and tube formation. So what about in vivo? So if you use the carbon tetrachloride model, I think I'm not going to show you the BDL model, but it's the same thing in both models, and you use neuropilin neutralizing antibody, you can attenuate the development of fibrosis as assessed by Sirius Red here. And you also reduce the numbers of PDGF receptor positive cells within these septae, as well as decreased amounts of the hepatic stellate cells expressing neuropilin within these septae. And again, if you look at a number of different biochemical and PCR-based correlates, just to add to the microscopy, hydroxyproline mirrors this, as well as PDGF receptor by PCR. And then just if in case, there's a number of other markers as well. So collagen 1, SMA, TGF-beta, and a number of others that all basically consistently show that you can attenuate the fibrotic response that occurs in carbon tetrachloride-induced cirrhosis with the neuropilin neutralizing antibody. And so one of the important observations that we observed there, and you'll notice that I started the talk talking about blood vessels and angiogenesis, and in fact, most of the effects we're seeing are fibrotic. And so in some ways we were a little disappointed because we're more interested in vessels than fibrosis, but as we're coming to the realization that the two pathways are quite tied together, nonetheless, this led us to look a little bit more carefully at whether neuropilin may have a direct anti-fibrotic effect as well by limiting stellate cell-induced collagen deposition. And indeed, we looked first at PDGF because that was the pathway we were looking at. Although PDGF isn't traditionally the most potent inducer of collagen from stellate cells, it does promote some collagen deposition, and especially once you overexpress neuropilin in hepatic stellate cells, and this is, again, the LX2 cells, you see that there's much more collagen deposition in a time-dependent manner. And if you use an alternative assay than Western blot, such as the Circol assay, you see the same thing, that cells overexpressing neuropilin make more collagen than cells that aren't overexpressing neuropilin. And you can see, again, it's not a very dramatic collagen response to PDGF because that's not the major driver in stellate cells for collagen production. And again, just not to forget about the c-Abl pathway, because it really, as you see, when you look at the blots in terms of what's the most potent, it's when you turn off c-Abl, nothing is happening. And so it's really quite an important step here. And again, as you see, when you take mouse embryonic fibroblasts that lack c-Abl, it totally turns off collagen production in response to PDGF. So one of the limitations in the fibrosis field right now is that when you turn off one pathway, there's other compensatory pathways that also can overcome. And thus, in humans, it becomes very difficult to actually induce some clinically significant anti-fibrotic effects. And in fact, the two canonical major pathways are PDGF and TGF-beta. And so those two together act to lead to this activation of these stellate cells. And so it's not really just the PDGF-induced migration that I showed you that's an important driver, but also the TGF-beta, which leads to proliferation of stellate cells, as well as the matrix deposition that I showed you. So because of that, we also looked at, and also because that's what our reviewers told us to do, we also looked at the effects of neuropilin on the TGF-beta pathway as well. And so what you see is, again, mirrors the effects that you saw in the PDGF responses, except overall there's just a more potent effect of TGF-beta on collagen production. But again, when you overexpress neuropilin, you have an increase in the amount of collagen production in response to TGF. And again, if you look at an alternative assay, using the Circol assay, again, you see increased collagen production in response to TGF-beta in cells overexpressing neuropilin. And again, with the MEFs, as well as the neuropilin knockouts, the neuropilin knockout MEFs don't have quite even as potent an effect as the c-Abl If I had to pick one target, actually, in retrospect now, I think c-Abl is really quite important, although clearly the neuropilin knockouts also have markedly reduced collagen production. I'm showing you data from a couple of very recently published papers. One of our collaborative groups was doing a lot of work as well in neuropilin, and they helped us a lot with the neuropilin reagents, and in turn, they worked mostly in endothelium, and we helped them with some of the myofibroblast work. And so what our collaborators and us together found was that when we knocked down neuropilin, there was also an upregulation of this protein ID1. And ID1, we discussed earlier, is actually not so much used in the liver field, but in the vascular field and the stem cell fields. It's thought to be a marker of quiescence. And this was interesting because while ID1 went up, again, we saw the reduction in SMA. This is very consistent with what I showed you before, a reduction in collagen, but an upregulation of ID1. And these are actually both downstream of TGF-beta, but they use different SMADs to achieve their function. So this led to some further experiments to try to understand how does neuropilin regulate the TGF-beta responses, and how can it lead to cell quiescence, upregulations of ID1, which is actually downstream from certain SMAD proteins, but downregulation of SMA, which is downstream from other SMAD proteins. And the answer, again, is a kind of a complicated Western blot I'm showing you, but just to summarize, is that when you knock down neuropilin, you actually see an upregulation of SMAD1/5, but a downregulation of SMAD2 and SMAD3. So neuropilin seems to be a switch that, in response to TGF-beta signaling, can shuttle signals towards certain SMAD proteins and not towards others. And this is shown more schematically here, where you see that normally TGF-beta will bind its receptor, R2, R1 phosphorylates, and you have downstream SMAD protein activation. And while SMAD2 and 3 lead to increased smooth muscle actin, SMAD1, 5, and 8 it leads to ID1. And this is more you can envision in the quiescent state. And when you overexpress neuropilin, or when neuropilin is functioning and acting as its net for presumably the same way through TGF-beta ligand availability, as I showed you for PDGF ligand, in fact, you activate the pathway that leads to SMAD2/3, increased SMA. Whoops, oh, went crazy there, sorry. Okay, there you go. So, so it increases SMAD2/3, increased SMA, and downregulation of some of the quiescence markers such as ID1. And so what about in human cirrhosis? This is just some correlative data using human cirrhosis specimens. This is in patients with hepatitis C. I'm going to stay away from the mouse this time so I don't do that again. And you can see that there's an upregulation of neuropilin. And again, you see the higher glycosylation band at 250 kD, and that's a glycoprotein. And that's the one that's particularly upregulated, and that's the one that we're proposing acts as this molecular net. Similarly, in these patient samples, you see upregulation of PDGF receptor as well, as would be expected. And if you pick a mechanistically different liver disease, such as NASH, you see essentially the same thing. Not much effects at stage 1 and 2, but by stage 3 and 4, you see an upregulation of neuropilin as well as upregulation of PDGF receptor beta. So this provides some correlation in human cirrhotic specimens as well. So this is sort of a summary slide of the potential roles of neuropilin in liver fibrosis. We propose that neuropilin acts as a co-receptor for key kinase pathways that are important in the process of hepatic stellate cell functions, including the ability of stellate cells to be recruited to the fibrotic milieu, for them to make collagen, as well as for them to promote vascular function. And downstream from these receptors is c-Abl, which transduces signals from PDGF and TGF-beta. I showed you some data with TGF-beta as well, and that leads to RAC1 activity. And RAC1 probably promotes migration and invasion. And although we didn't show that here in terms of collagen deposition, other groups have shown that RAC1 is also important for NADPH oxidase activation and other pathways that lead to collagen deposition as well. So this is a cartoon, or this is a micrograph. Actually, this is from a rat model of liver cirrhosis. And what struck us was that when you stain these for alpha smooth muscle actin, In fact, the pattern is really quite similar to what we see with cirrhosis. You see these nodules of hepatocytes. Granted, they're malignant hepatocytes and they have mutations in their genetic makeup, but you see a dense desmoplasia around nodules of hepatocytes, and high vascularity as well. You can see some of these very prominent SMA-positive vessels as well. So it led us to this idea that there may be some parallels in cancer and cirrhosis, especially in tumors where you have a great deal of desmoplasia. And in fact, although we don't really work in cancer per se, we don't do anything with epithelial cells, but we are interested in the microenvironment, and we think that the tumor microenvironment can be really quite important for tumor growth. So what do parasites do in the tumor microenvironment? Well, one is that they activate into myofibroblasts, and again, that sounds familiar because that's the same thing that's happening in cirrhosis. And they deposit matrix. This is the desmoplasia. We don't use that term so much in liver fibrosis, but in cancer, it's really a prominent desmoplasia that you see. And so this is the matrix deposition from myofibroblasts. Parasites, as I mentioned, they also mediate angiogenesis, and they add durability to vessels. And so this is the idea that these parasites are coming into the vessel. And then they secrete growth factors that regulate angiogenesis. So again, VEGF is secreted by myofibroblasts. That recruits endothelium to the tumor microenvironment. And then they also respond to endothelial cell-derived growth factors, as I said, like VEGF. And then they make also a number of factors. And so, so there's this intricate crosstalk and interplay, not just with the tumor cell here in yellow, but with the endothelial cell and the parasite, which can transform into a myofibroblast in this tumor microenvironment as well, especially with the idea of matrix stiffness and the idea that stiffness of the matrix can be an important driver of increased malignant potential and metastasis. There's probably an increasing role now of the myofibroblast in the tumor microenvironment that previously wasn't as well recognized. So this led us to ask some questions about what neuropilin might be doing within the tumor microenvironment. And here again, I'm going to show you another signaling pathway, but some similarities, but some unique aspects as well. And none of this is published now. The last data sets I showed you were recently published. And this is to test the idea whether neuropilin may also regulate another very important signaling pathway within myofibroblasts, which is the integrin signaling pathway. Now, integrins do a lot of things for cells, but one of the important things that they do within the tumor microenvironment is that they convert soluble fibronectin into a matrix-bound insoluble fibronectin. And why is that important? That's important for many different reasons, but one of them is that once you have this fibronectin coating, it allows increased cell adhesion, but also seems to be one of the important steps that allows subsequent collagen deposition and increased tumor stiffness. So we've been particularly interested in how neuropilin could regulate this conversion of soluble fibronectin into insoluble fibronectin, since this is one of the important functions of the integrin, especially alpha-5 beta-1 integrin. So again, we went back to our neutralizing antibody, and here I'll show you some in vivo data using the neuropilin neutralizing antibody. This is from Genentech. And in fact, you know, when we speak with the company, they're using the antibody the most. Their biggest indications are in cancer rather than in cirrhosis. Actually, they really don't have much interest to pursue this in cirrhosis. It's, you know, again, it's a company decision, but they have a lot of interest for this antibody in cancer. And what we see here is, and these are subcutaneous models, so this is not within a liver tumor model per se, but in subcutaneous models, if you take LLC cells, which if you put into C57, since LLC is a C57 black 6 tumor line, you see that when you use the neuropilin neutralizing antibody, you attenuate the tumor volume. And in fact, when you look at fibronectin fibrillation, staining with a fibronectin antibody, there's a marked reduction in the amount of fibronectin. I didn't show you here, but there's also reductions in angiogenic markers and markers of myofibroblast activation within the tumor microenvironment. And this is just quantified here, showing a reduction in the amount of fibronectin within these tumors. Now, here, when you give the antibody systemically, that doesn't really prove that the effect is on myofibroblasts, because tumor cells, as I mentioned, also express neuropilin. So we took an alternative approach here, and this is with a co-implantation model. So here, this is kind of an interesting thing with tumors that when you do your co-implantation, if you take some myofibroblasts and put them with the tumor when you do the co-implantation, why the tumor grows bigger. And that, again, just supports the idea that the myofibroblasts are very important for tumor growth by virtue of their ability to release matrix, which gives the tumor cells a place to adhere, and their ability to make growth factors. And in fact, if you look, this is just some data from another study that we did, and if you label these myofibroblasts in the co-implantation, you can see that while they actually form very nicely in vessels and coat the vessels and integrate into the stroma and amongst the tumor cells. And in fact, this is what I showed you, that if you take tumor cells and then you co-implant them with mouse embryonic fibroblasts or other myofibroblast precursors, you can increase tumor size. So using that model, here again, this is the LLC model, so taking tumor cells, and again, we go back to our neuropilin flox mice here, and if we take the myofibroblasts, transduce them with Cre to knock out neuropilin, and then overexpress, and then do the implantations in our host. And in this case, the host is the neuropilin flox mice crossed with SM22, which again knocks out neuropilin from myofibroblasts within the skin and the areas where you're implanting these tumors subcutaneously. So now you've knocked out neuropilin from the host myofibroblasts as well as from the implanted myofibroblasts. And again, you see this reduction in tumor volume. So what's the mechanistic basis here? It just shows that we've reduced tumors, but I really haven't shown you a lot of information about how it regulates fibronectin fibril assembly. And I'll show you a little bit of this data, although this is, as I said, is still pretty much a work in progress. But if we really want to study fibronectin fibril assembly, we need to use an exogenous biotinylated fibronectin, because that's the way that we can exclude increased fibronectin production in the presence of neuropilin. So that's indeed what we did here. So we overexpress neuropilin or overexpress RFP, which is a control. And then we look at the fibrillation of exogenous biotinylated fluorescent fibronectin, and you can see that there's an increase in this kind of reticular pattern of fibronectin consistent with a fibrillated fibronectin. And similarly, if you knock down neuropilin or the beta-1 integrin, which is the driver of this process, you see an attenuation in the amount of this fibrillated fibronectin that you see here. And these are just some siRNA controls. And similarly, if you take these mouse embryonic fibroblasts from the neuropilin flox mice and remove the neuropilin, either by crossing with SM22, which knocks out neuropilin from mouse embryonic fibroblasts, you see the reduction in fibronectin fibrillation. And another assay to look at fibronectin fibrillation is this solubility in deoxycholic acid, which insolubility in deoxycholic acid basically defines fibrillated fibronectin. And again, you can see that there's an increase with neuropilin. And these are a couple mutants I'm going to define for you in the next slide. But you'll notice that when we mutate a couple of these sites in neuropilin, which again I'm going to show you on the next slide, you see some reduction in this neuropilin overexpression. So this starts to get us to a mechanistic basis by which we can start to identify which portions of neuropilin are mediating which functions. There's the cartoon. So the two sites that we're mutating, and I'll show you a little bit of data with these mutations. We haven't deciphered them fully yet, but we're on our way with this. One is at serine 612, and this is an important site for glycosylation of neuropilin, and is probably the dominant glycosylation site that makes neuropilin into a proteoglycan. And the other site, this is a deletion mutant, not a point mutation. This is the C domain. This, again, is just about 40 amino acids that are on the intracellular portion of neuropilin, and these mediate a lot of the signal transduction that comes through neuropilin. And what I show you here is that neuropilin does indeed bind in fibronectin, similar to what I showed you with PDGF ligand. And if you immunoprecipitate with this biotinylated fibronectin that you add, you can pull out neuropilin, especially if you overexpress it. And if you go straight to the IPs here on the far part of this panel, again, you see a very prominent increase in binding. This time we IP with neuropilin instead of biotinylated fibronectin. And again, both of these mutants have a prominent reduction. What we're really trying to decipher is how these mutants are acting. So again, you can envision that the 612 mutant is blocking out that net, right? So I showed you the idea of neuropilin have a glyconet, and the glyconet pulls in these ligands, perhaps in some degree non-specifically, but can bind lots of ligands that are important for stellate cell activation. And maybe we can knock out that net with 612. And the C domain is what we think is actually mediating intracellular signaling and interactions with c-Abl and such, although we haven't actually been able to show yet that this C domain binds to c-Abl. This is certainly thought to be the canonical intracellular signaling domain of neuropilin. So again, how does neuropilin promote this fibril assembly? It binds the fibronectin, but what is it actually doing to integrins? Well, what our data shows is that it's actually activating integrin function. And so again, this is just to show you that indeed this is integrin dependent. So if you knock out integrin, neuropilin won't do anything to fibronectin fibrillation. It requires the key steps that integrins mediate for stretching, pulling out that fibronectin, increasing cell tension, and then depositing the soluble fibronectin into a matrix. And indeed, if you look at Hutt stain, which is one marker of activation of integrins, you see a prominent increase in activity of integrins, alpha-5 beta-1. And also, I haven't shown you here, but if you use a GSD fibronectin, which just has the domain that binds to the integrin, you see an increased amount of binding to the integrin, suggesting that the integrins are getting activated when you overexpress neuropilin. And again, c-Abl seems to be playing an important role here again. So if you take these MEFs that lack c-Abl, again, you see much less biotinylated fibronectin fibrillation. And this is also shown by the DOC insolubility, where the c-Abl knockout cells have much less fibrillated fibronectin when you add biotinylated fibronectin. And again, just to take it back to the human level, and I'll be wrapping up here. This is getting near the end here. If you take this back to the human level and you try to correlate neuropilin expression in tumors, these are all hepatocellular cancers. And what you can see is that the amount of neuropilin correlates with survival. So patients who have much higher amounts of neuropilin, here we're looking at the upper quarter versus the lower quarter. If you just take the median, it doesn't correlate. So some of you who really know these methods probably better than me would know that, well, you may ask that. So what we see is if you really look at high expressors or low expressors, that seems to have quite an important effect in correlation with patient survival. So this is a cartoon that kind of shows the proposed mechanism of neuropilin and how it regulates fibronectin assembly and tumor growth. So again, we think the glycodomain is important here. I showed you that data with the S612 mutant, which anchors a lot of the glycosylation of neuropilin. And we think that the intracellular domain is also important. And we're starting to realize that there's probably some important allosteric regulation between the intracellular regions of neuropilin and the extracellular. And that may be why we see very similar phenotypes with both mutants. But right now, we're still trying to ferret that out, where we can dissect which mutant will block what, but that it won't block something that the other mutant's blocking, because right now both mutants are just blocking a lot. And so we think that the neuropilin mediates the enhanced fibronectin binding to alpha-5 beta-1, and that's at the extracellular level. This increases fibronectin fibrillation. The c-Abl pathway likely leads to increased integrin activity. So you have increased activity, and you also have increased bioavailability of fibronectin. And these two together increase the fibronectin fibrillation. And we think that this enhances the overall tumor growth and thereby contributes to what we term as an active tumor microenvironment. So I just want to summarize what I showed you, a lot of data, but hopefully some of the cartoons could put it into a big picture way. And we talked about liver fibrosis as well as cancer, but really I think as many of you would agree, it's really a parallel between fibrosis and the development of cancer. And so what I showed you here was that neuropilin promotes signaling pathways in hepatic stellate cells that lead to myofibroblastic activation, angiogenesis, matrix deposition, and fibronectin fibrillation. And we think that this may be likely to contribute to myofibroblast-related pathobiology in both liver fibrosis as well as cancer through effects on the tumor microenvironment. And so there I'm going to conclude. I'm going to thank a number of the people in the lab who worked on this project. Sheng Cao and Usman Yaqub drove many of the experiments. Dominique Thabot also worked on a number of experiments, not in this paper, but in other projects that were related. And some of you may know her, so I'll just mention her there. And with that, I'm going to conclude and take questions.